And this year we're also planning to conduct more hands-on training sessions also for various human relevant technologies, hopefully also computational. So
Good.
that's going good, and our team is also growing. So now we have 3 more members. So yeah, probably you might have gotten mails from them. So Kasturi, who's the senior research associate, and Anushka is the research associate, and Kadambri, she's the junior research associate. So all of them are like very good psychom and biologists. So yeah.
Yeah, that's good progress, I think.
Yeah.
You're doing good.
Thank you.
So this is the your organization there. Huh?
Yeah, so this is Atal Incubation Centre. Like there are a few. So we are a part of Atal Incubation Centre. So
Sure.
yeah. So they have many other programs also. So yeah, we are one of the programs. So although our work is kind of slightly different because it's basically an incubation center. So but our work is kind of slightly different because we don't host startups. We are more of a policy center.
నేను పీడి సార్కి ఒక ఫోల్డర్ పంపించిన దాంట్లో 1 2 3 4 5 అంటే 4 ఎఫ్ఏలు ఒక SA1 టోటల్ చేస్తే
Can you mute
మన ఫైల్ వేసి
So you got the administration also there?
Sorry?
So you got the administration staff there along with your research team?
Yeah. So we have like a core Atal Incubation Center team where where we have administrative people also. So
okay, good. That is very good actually, I think.
Right.
So participants are belonging to like students or faculty or
So we have so we have like a good mix of different populations, like because from our registration we kind of know what is the population of various. So we have 30-40 percent will be BSc students, and then we have MSc students, PhDs, but also researchers. So we have like a good mix of different sections, but majority would be, I think, BSc, MSc, PhD students. I think 50-60% will be that. But then apart from that, we'll have maybe 30% also PhD students and higher up.
okay. So overall, the ambition of this webinar series is to like provide the knowledge gain from the expertise group to the participants.
Right. Kind of bring more awareness for various different. So initially we had a focus on organoids and organ-on-chip and human relevant this thing. So almost for since last February, we have been focusing on computational and predictive modeling kind of thing. So we've had speakers speak about different aspects of predictive modeling, computational biology, omics, genomics, predictive biology. So different aspects of that. How do you model, for example, drug development and all of that. So just mostly so almost for this last 7 or 8 webinars, they have been purely focused on the computational aspect of it.
Okay, good.
I think we can go ahead and begin. So I think mostly everyone should have joined by now.
Yeah, sure.
Okay.
Hello, everyone. Good morning. Today we have the 22nd edition of our webinar series. I will start with a short introduction to our center. So the Center for Predictive Human Model Systems is a science and policy center and a think tank established in 2019. Our focus is basically to enable new technologies and model systems which are based on human biology, like organoids, organs on chip, 3D tissues, computational biology, and so on, rather than primarily relying on animal models. The main aim is to prioritize investment and research in India on these technologies. So far, we have published 3 white papers on developing adverse outcome pathway frameworks, developing systems biology research, and using MPS as a model system, all in the Indian scenario. We conduct monthly webinars as a part of our education and training, as well as increasing awareness sector. And we have published several popular science articles, and we've conducted an India EMBO lecture course. We also do policy initiatives where we've published the 3 white papers I mentioned. We've conducted 2 multi-stakeholder roundtable meetings, and we are prioritizing investment and regulatory approvals in these methods. So we plan to enable human relevant research in India, and these are certain steps we are taking forward for that. And today we have Dr. Feroz Khan, and he's the scientist working at the CSIR-Central Institute of Medicinal and Aromatic Plants in Lucknow, India. And Dr. Khan holds a PhD in plant biotechnology and a PDF in pharmacology. He is known for his research in the field of plant biotechnology, particularly for his contributions in the area of plant genetic engineering and functional genomics. Today, he will be delivering a talk on the topic of 2D and 3D QSAR models on drug development, which highlights his research in the area of computer-aided drug design. Welcome, Dr. Khan.
Thank you, Kadambi. Thanks for nice introduction. Yeah.
Thank you.
So can I
So
start
we can,
the presentation?
yes, sure.
Okay, thank you. Are you able to see this presentation?
Yes, we can see.
Okay. So welcome all the participants in this webinar series organized by Atal Incubation Centre, CCMB, coordinated by Dr. Surat and her team members. So today's talk is on like 2D and 3D dimensional QSR studies related to drug discovery domain. So my topic I have chosen based on that area only. That is 2D and 3D QSR methods in drug design and discovery. So let's start with the introduction part. So if you see the drug discovery pipeline, the start mostly the research component start with the lead identification and optimization method, and during the process of like up to the preclinical trials, the flow chart is already given in this slide. Actually, you see, we normally start with QSR, and for that we require data set from experimental activity.
हम लोग कैसे पता करें उसे पुराने वाले उसका कौन सा
Hello.
Sorry.
Please mute your mic.
Yeah. Anushka, please.
आज भी नहीं हो पाएगा
सारा नहीं करना एक ही दिन में तुम एक ही दिन
Just 1 minute. We request everyone to keep their mics on mute. Thank you.
Okay, so let's start with the like introduction part. So for lead identification and optimization, we required quantitative and qualitative QSR models, and the name itself indicating there is a like prediction model, and they relate the biological activity with the chemical properties, especially in 2D QSR. While in the 3D QSR, normally we take with the alignment features from the structural properties of the chemical structure and the receptor molecules, and that component information is taken for further forward for mapping the different ligands to predict to develop the model for predictions. So both the approaches would work, and for this we required QSR modeling step in the initial step, especially before the like chemical synthesis of the molecules. And beside QSR model, there is another approach called as pharmacophore modeling that's come under 3D QSR approach. And with this prediction model, we predict the lead, potential lead, and these predicted leads are further synthesized by the chemist and then go forward for in vitro studies. There are so many screening studies also in along with this QSR approach that already like discussed by the research group earlier. and webinar series given by other experts. But still, I will like to inform some of the screening strategies. So normally we go for screening strategies for drug likeness and lead likeness studies and target identification and validation and admit analysis. And after that, we go for preclinical or clinical studies on small animals or human clinical trials too. You see, the definition of the QSR is a process by which biological activity is correlated with physicochemical properties, and this relationship expressed in the form of mathematical equation, and this equation generated is called as QSR model equation. It's a mathematical relationship where normally we correlate the biological activity with chemical structural properties. And this normally, since the data set is not so much high, so normally it's a linear relationship basically. If you see the worldwide regulation about the QSR, US FDA already given the instruction on their website that all the developer of related to drug discovery domains must submit the QSR details before approval of any compounds. So you see, go to the regulatory reports of the US FDA and find out the regulatory concern related with QSR study. We have mentioned some of the softwares to how to develop the models, especially the TopCat, MDL, QSR, multi-case. I already highlighted in the uncircled regions. So you just go to this website and see more detail about the QSR approval before processing for clinical trials. Similarly, similarly, uh, similarly, other European group also provided some guideline for discovering lead identification and exploration of mechanism of action of the drug-like molecule to submit the QSR model. And this require 4 components. If you see the REACH guideline of the European group, they already mentioned in their website, this is already given in the this presentation slide. You can go and see more detail about the different part of the model development validation. The major 4 component are results are derived from QSR model whose scientific validity has been established. You must submit this information, and after that you have to provide the information related to substance falls within the applicability domain of the QSR model, and then results are adequate for the purpose of classification and labeling and risk assessment. And last component is adequate and reliable documentation of the QSR model is must provided for the approval. So European group also already like in FDA, they provided the guideline how to submit the QSR model information of the drug-like molecules. So if you see the introduction part or the historical part of the QSR, how it developed basically. So in this slide, I just mentioned that the Professor Corwin Henge basically first time relate the biological activity with the properties and first time developed the QSR model in 1969. That equation is already mentioned here, and it relates the concentration with the log P value. So log P is a physicochemical property relating the bioavailability of the molecules through intestinal membrane. And apart from that, he mentioned some of the constant which are basically used for a static purpose. So some of the information related to 2-dimensional and some of the components are related to the 3D components. So log P, just like log P in 2D-dimensional 2D QSR, log P is a very common property used for relating the biological activity. So what do you mean by log P? Log P is a measure of the drug hydrophobicity, which was selected as a measure of its ability to pass through cell membrane. So it's mimic the property of absorption of the molecule from intestinal membrane to blood serum. If you see the structural properties, structural file of the molecule, it comprises the information related to 1-dimensional and 2-dimensional and 3-dimensional information. Similarly, after the molecular properties we have to like extract out, you should submit the molecular descriptors required for modeling. And these are some of the common descriptors category which are used for extracting the information of the molecules through molecular structural profile. And these structural files are either present in the form of CDX extension or SDF file or PDB files. And there are so many other file extensions which are related with different specific softwares, but most commonly is CDX, 2D preliminary structural file, and the database is based structural file in SDF. So you will find these constitutional, electronic, geometrical, hydrophobic, and lipophilic solubility related properties. Calculate these properties, and it's around like from one single molecule, you can predict the properties related with 100 to 100 and thousands properties. These properties you can like go for further for pre-processing, and pre-processing under this category we provide normally normalization, standardization, and feature selection and outlier detection. After cleaning of the molecules properties and the molecule itself, we normally go for relationship studies, and this required multivariate analysis. For this, we use multiple linear regression, self-organizing map, principal component analysis, partial least square method, neural network, support vector machine, and after that, we evaluate the model through R square or uh, validation square, error square, and then plot the data on the graph, basically comparing the predicted activity with the experimental one. So what do you mean by molecular descriptors in QSAR? Basically, it's chemical properties in quantitative manners. These are the numerical values that characterize the properties of molecules. These are the uncoded structural features of molecules as numerical descriptors. And these properties you can calculate through open source free softwares, and it requires some time for calculation of different properties. But after calculating two-dimensional and three-dimensional properties of the molecule, a small molecule, I'm talking about the small molecules. So you can like get thousands of properties for a single molecules. Now you have to relate these properties with the biological activity, and this relationship is basically you require number of methods, statistical as well as machine learning methods. So once you like wish to calculate the chemical properties from the molecule file like SDF or PDB, you require to extract the features related with number of bonds, number of atoms, number of other components. Similarly, there is information about the vector quantities related with the molecules and the functional component within the molecules. So within a single molecule file, you will find number of things like single bond, double bond, triple bond, molecule information, physicochemical properties, and fragments related information. So these basically information is already given in the two-dimensional SDF file or PDB file, but you require to calculate number of properties through using this information from other third-party softwares. Even commercial softwares are also there. Like Paddle software is very well commonly used by the research group or the students. It's a freely available open source software. You can just load the molecule and it predict the different 2D and 3D properties within a like a minute or 2-3 minutes it takes. Once you open the two-dimensional structural file in ACD file related or ChemDraw file, you will find that there is information of the table in the form of like matrix. There is information about the first component, like 1 to 3 lines are basically related with the introductory part, or it is also called the header, while the rest of the like 4 to 17 or 18 lines concern the connection tables, which is a required information about the atoms, bonds, and their bond length also. There are 5 different categories within the chemical descriptors or the properties. You must include these 5 descriptors within your model. Before making a relationship, you must calculate it. So what are these 5 components? As mentioned here in the slide, you see constitutional descriptors are the number 1, which are normally control the biological activity. And the second is electrostatic descriptors. You must include the charges, surface areas, And the third most important descriptor is topological descriptors, which are related with topology of the molecules. It includes the valency, connectivity indices, and so on. And lastly, geometrical descriptors and quantum chemical descriptors. So those properties which are related to geometry, you must include these properties. For example, solvent accessible surface area I mentioned here, volume or molar concentration related volume, and the quantum chemical descriptors like related with orbital energies, HOMO energies, dielectric energies, and so on. So once you include these 5 categories in your QSAR model equations, then I'm sure you will get the good relationship model. I've just shown the snapshot of the chemical properties once it is calculated for number of compounds. You see the rows indicates the compound number and the column indicates the different chemical properties. So I listed some of the calculated properties. It is calculated through paddle softwares. You can use other softwares if required. So each column indicate different chemical properties. For example, first column indicating the title of the molecule, and the second column onward there is a property related things. Log P, for example. So you see the second column indicating the different log P's values of different compounds, and so on. Once you calculate the property, you must calculate the correlation between the chemical properties and the biological activity, so that you can find out the different important chemical properties which having the relationship with the biological activity. So those chemical properties which shows no correlation, you can exclude that and only include those which have correlation value more than 0.5 or 0.6 to 1. So in the range of 0.5 to 1, you can include these correlation data and these chemical properties in your relationship. I'm sure you will get the good relationship model after that. So what is the process of making the QSAR model? If you see, the first part is to develop the training data set. So training is required in this modeling step because it's a supervised method of machine learning. So you must provide the training. And what do you mean by training? Training means already reported activities and the reported compounds. You must normalize these activity in the form of log values, and then relate the biological activity, normalize biological activity with the chemical descriptors to make a relationship in the form of line equation, which is also called as since there are so many multiple properties. So that's why we use the term multiple linear regression. So when we go for prediction, we always use the regression model, and this regression step is required during the relationship. And if the data is very much high, you can use the machine learning method like SVM, ANN, and so on. So second step is to calculate the chemical features, properties, and these chemical properties also called as chemical features. You calculate these 5 categories as I mentioned earlier, and find out how many properties you have correlated with the biological activity. Divide the training data set into 2 groups. After that, once you calculate the properties, divide the training data set into 2 groups. 80% part should be included in the QSAR model development part and 20% part included in the test set. So second set is test set, which is used for model validation further. And this model validation test set is actually, it's also called as external validation step, because these are not the component of the internal model development process. And lastly, the query set. The third is query set. Once you develop the model, you wish to predict the biological activity of your untested molecule or unknown molecules whose activity is not reported. So before going for clinical trials or preclinical studies, you must calculate the biological activity through this derived QSAR model and see whether your compound, untested compound, having the possibility to be a drug-like molecule or not. So this way you can classify the number of virtually designed molecules through QSAR approach. It's a very good approach nowadays, and earlier it is also used for making the mechanistic QSAR model to reveal the mechanism of the lead molecules, why it is so much active. In this slide, if you see, I have just proposed the like how to make the model graphically. I just try to reflect the step of the modeling where you require training data set. Training data set must be like more than like 40 and 30 compounds, not less than 30, because it will statistically give the error then. So the compounds, known reported activity compound should be more than 40 is good enough. And you can include a number of compounds if you have in your database or your selection. So 100 is good enough to make a relationship. So training set include that component. And after that, you calculate the properties, which are also called as parameters, chemical properties or descriptors. And after that, make the relationship. In the middle, you see the uncircled part. I just mentioned what is your aim actually. If you want to predict the biological activity, go for regression. If you want to classify the compounds only based on like active or inactive compounds, you can like use a classification model here. Depending upon the need, you can use any machine learning approach or a statistical approach to make a relationship. But 90% people like in research area uses the regression method to make the relationship and give the prediction for untested compound further. So it requires test set, external validation set, and unknown query compound set for prediction. And then we got the predicted activity. And this way we can screen number of compounds. So why QSAR required actually? So as you know, in vitro assay, we normally got the bioactivity information of the small molecule. But in terms of some IC50 or KI value or KD value or MIC, but we do not get the information about why it is so much active. So this information you can like why it is so much active, this information you can extract out through QSAR approach. So this problem is basically a mechanism revealing step. QSAR is reveal the biological reasons, biochemical reasons why this compound is so much active, and what are the chemical properties regulating this biological activity. So out of thousands of molecules properties, you can ultimately find at the end that there are only 4 or 5 properties are basically controlling the biological activity. And these chemical properties will come in the form of equation, line equation, y is equal to mx plus c. You will find that these properties showing the different strength for regulating the biological activity in terms of coefficient values. So it's a mathematical relationship. So you just find the relationship in the further slide. I will show you the example. And the second important part is to predict the activity of untested compound before going for chemical synthesis and wet lab testing, which required high amount of the money for testing. So before like going for testing, you must include the predictive biology of this approach QSR to know the whether the compound is active or inactive. So if there is a possibility that it reaches the biological activity in the range of like 60%, 80%, then we normally go forward further to predict the biological activity through in vitro tests. So once you develop the QSAR model, you can predict the biological activity and then go for chemical toxicity predictions and then go for environmental activity related, toxicity related information you can predict the molecular properties. And ultimately, the most significant component or utility part of the QSR is it saves the wet lab economy and time. And the last most important is save the life of a small animal. So it uses before preclinical trials to prioritize the lead. So it is very commonly used nowadays in all the research lab, even the pharmaceutical lab already using from last 50 to 60 years. So that's the beauty of this QSR approach. Some of the example, if you see commercialized drugs in the market derived from classical QSR approaches, metconazole, lombrazine, blufofin, and norfloxacin antibiotic, and so on. There are so many examples where the structural component is mechanism related information is revealed by QSR approach. And I just mentioned some of the slide here for your example information that these are the drugs derived in 1983, 1975, 1984 by different pharmaceutical companies like Bayer's in 1975 developed the metametron, that is a sugar beet herbicide, first time through QSR approach. So it's a very approved, classical or old approach and very well related with chemistry, very grounded chemistry. So without the much error actually. So most of the chemists rely on 2D QSR. This is also called as 2D QSR because it relates the chemical properties with the biological activity. So what are the disadvantages of the QSR? We do the if you have a less knowledge about the mathematical component as well as the detection of chemical properties, you make a false correlation, and this false correlation gives the error in the end while predicting the biological activity. And the second component is identification of the training data set, which is normally noisy data set, normally available in the form of Campbell database or PubChem and so on. So you have to clean the data before developing the model. And normally this step is erroneous. Most of the research group do the error while selecting the training data set. And that's why it gives the wrong correlation or QSR equation. And the third most important is calculation of the right chemical properties which relate the biological activity. So normally we provide the 2D dimensional properties to relate the biological activity, but there is a need if you have a 3D structure file, you just extract 3D structural information of the ligand and include that properties also in the QSR model. So 2D and 3D information is required for good QSR model. Otherwise it will give the error. So it requires some of the knowledge of the chemistry and some of the knowledge of the some statistical component or machine learning related things. And I suggest you just collaborate with chemists before developing any QSR model. Virtually developing a QSR model without input of any chemist or biologist, it give always error at the end and whole exercise gone waste. So don't go alone, make a team and then go forward. So now we see the example of the QSR model, 2D and 3D both. So some of the snapshot is just revealed here in the slide, where the lead molecule, which is shown in green color, in bind at the binding side of the protein target, and having the some bond land interaction, which is also called as hydrogen bond interactions between the molecules, as denoted by yellow color dot. So those molecules which have the hydrogen bonds interactions, they are likely to have a drug-like properties. So this slide shows the 2D QSR equation, how it is represented. So for your example, I just taken the snapshot of the training data set as well as test set as well as query set compound. Not all, but some of the component I've taken it. And you see in the table, the first 4-5 rows indicating the known drug related with anti-inflammatory drugs or immunomodulatory drugs. And these anti-inflammatory known drugs you must include with the biological activity. I have taken the LD50 and converted in the form of log LD50 and then develop the relationship. So relationship is already given here in the graph form. You see the experimental log LD50 and predicted log LD50 is in the linear line, and they have high correlation, which is also called as R square, which is a regression coefficient value, which shows that 0.99 correlation we found at the in between the known drug and the biological activity. So you see, I found number of chemical property, 5-6 properties correlating the biological activity. So these chemical properties already mentioned in the QSR equation at the end. You see, C is a concentration and K, R, I, AI and AMA are the chemical properties as mentioned in the right side at the end. So C, K is basically dipole moment which regulate the biological activity, and steric energy is represented by R, and T is representing the group count, which is amide, and the AI means lambda max UV visible in nanometer unit, and AM means molar refractivity. So these are the chemical properties which regulate the biological activity. So you see some of the chemical properties are having the negative correlation and some of the positive correlation. And RCV square, which is also called as cross validation regression coefficient, which is also very good actually, 0.96. It should be more than 0.6 actually. So whenever you develop the model, regression value should be more than or equal to 0.6, and cross validation or test data set regression value should be more than 0.6. So it is under the limit. So we have like published the data, and it is already in JMM journal. You find it. If you wish, you can explore the Google for more information. This is another example where I developed our group developed the QSAR model through following the OECD guidelines, and we followed the uh, different statistical parameters given by the OECD. And here we have taken 28 training dataset compound, and 6 we uses the in test set, while 4 properties, we chemical properties will finally be concluded that these 4 properties regulate the biological activity. And we found that predicted activity and experimental activity at the end is equal almost. And you see the regression plot, it is in linear fashion. Red color dot indicate the training data set, which is the experimental molecules with activity. And the blue dot indicates that these are the test compounds, but they are not included in the model development part, but they are the known experimental data molecules. So both blue and red color lines are basically in the dots indicating that they are both interrelated and diagonally arranged. So it gives the idea that they have a good relationship. And you see that these chemical properties in the contribution plot, 2 properties have positive correlation. These values are represented in the form of contribution in terms of percentage. More than 0 value in positive are basically positive correlated, and below 0 is in minus, they are called as negative correlated properties. So 2 bar indicating negative correlation properties and 2 bar indicating positive correlation. The bar in high length basically shows the more significant component of the model. So out of 4, the first chemical property, which is represented as nitrogen count, SSN count, which is also called as nitrogen count, in the chemical compound is must for biological activity. So at the end, we reveal the mechanism why it is so much active through chemical properties, and what are the chemical properties regulating the biological activity. And after training set mapping and test set mapping, we found that there are actual overlapping of the blue and red color line, and red color line indicates the actual data, while the blue color line indicating the predicted one. So both are overlapping 100%. It means they have a good relationship model. And this model is already published in PLOS journal. You can explore it in Google for more information. Now these are the positive model. Now let's see the error related model. It looks very similar, but easily it is not identifiable because it also shows the like diagonally arranged training data set and test set. But even the R square value and Q square value is almost very good, more than 0.6. But if you see the mapping part, where the overlapping of the training data set and the actual data set is not mapping actually. In the training data set, it is showing somehow good, but test data set overlapping graph, you see, they are not overlapping. Red color line are not overlapping with blue color line in the circle. So this way, this model is not good enough. You can reject it and go further for further revision model. So this way we can identify the error-based model also. This is example of error-based model actually. Yes, another example I just mentioned here where we have taken a very high number of known anti-cancer compounds from the database, and we use the NCI database of USA and extract out 2,291 compounds having the anti-breast cancer cell line activity against MCF7. And you see, since the number of compounds, since experimental compound is more than 100, so we use the machine learning method. So machine learning method we use first time here is SVM, which is also called a support vector regression, and develop the relationship between the 2,291 compound with their anti-breast cancer cell line activity. So finally, we come out with the R square value with 0.88 and Q square value with 0.87. And we use the cluster algorithm or since the number of compound is so much high, we have to like classify the compounds because they are structurally very different with each others, but some are similar, but most of the like compounds are in different groups. So we clustered the compound, these 2000, more than 2000 compound into different clusters first, and these finally we come up with the 8 clusters, and these clusters represent centroid molecule on the top. So if you see centroid molecule structure, all are having very different structures. So structurally they are very different, but biological activity is common and same. So that is the beauty. So it means there is no correlation that the structurally similar compounds having the same biological activity or vice versa. So that's why we require to do the mechanistic QSR studies. Now, after developing the 8 cluster, we developed the QSR regression model. And for each 8 clusters, we developed almost say 7 QSR model with good accuracy, while the model number cluster number 6 model is not good enough, as highlighted in the table with red color marks, because it shows the NP ratio less than required one, that is 3.0. So you must include the number of compounds more than 30. As I mentioned, in cluster number 1, there are 61 compounds, and in 2, 43, in 3, 70 compounds, and in 4, 47, in 5, 67. While in the 6, cluster number 6, there are 15 only. That's why we have not considered it. And number 7 and number 8 having a 56 and 38 compounds in that. And their R square values ranges from 0.7 to 0.85. So highest is 0.88 actually, as shown in green color. So model number 7, that is a cluster number 7 representative. It is the highest best one model which predict the activity. Now the next part is to make the relationship in the terms of graph. You must develop the graph first. If there is a relationship, then there is a diagonal line indicating the relationship. If the line indicated horizontally, it means there is a no relationship. So you see there is a diagonal line which indicating the positive correlation between the biological activity and the experimental chemical properties, or the model is good enough to predict the biological activity similar to experimental one. Now in this table, if you see the experimental and the predicted activity is almost similar. Once we develop the model and go for predictions, we found that all the internally used compounds having a good predictions accuracy, and they predict the biological activity, that is GI 50 value I used, that is a growth inhibition 50% population in molar concentration, and we normalize in natural log form because there is a biasness in log base 10, so we normally use the natural log for high number of compounds. So I suggest use the natural log if the compounds are so many high. So this way we find out the overall QSR model and like use this model for query set predictions. So what we done, we use randomly taking the phytomolecules, and that is, if you see the screen on the top, we randomly use the molecules from the plant molecules, and match this structure with the 8 clusters, and we found that cluster number 7 basically having the match with these compounds as shown by red color arrow. And after using this model number 7, we predict the value with R square value 0.95. And after prediction, we have to decide whether it is inactive or less active or mild active. And we found that predicted activity is almost similar with experimental activity. That is, even you see minus 6.72 in log form, natural log form in GI 50 value is similar to experimental one. So this is the beauty of the model, that based on its structural features, we select the respective model and then go for prediction. So this is another approach for high throughput screening method. This method is already published. You can explore in the Google and find more information about how to develop it and use it. This is an example of 2D QSR model where we have to develop the QSR model for oxalic acid in CMAP, Lucknow. And oxalic acid basically is a terpenoid plant molecule series which is having a good activity against cancer. cell lines. And after prediction, we gone that that in vitro evaluation shows similar kind of the results which give the predictions models. So this prediction model is good enough, QSAR model, to predict the biological activity of uracilic acid derivatives or the analogs against lung cancer cell line as well as CNS cancer cell line. So likewise, we developed so many QSAR model in CMAP and published in very reputed journals. You can go on through. And in this journey, I developed three in silico assay, three method in silico, and rest of the QSR and docking based models for different plant molecules like uracilic acid derivative, camptothecin derivative, brevifoliol derivative, endoyl chalcones derivatives, and glyceric acid derivatives. Mostly, most of the compounds under the category of terpenoids and chalcones and flavonoids. So you can also use this approach for virtual screening. And one of the method I used in CMAP for screening of the phytomolecule based on Gomberg's function assay. It's a in silico enzyme assay we developed for detection of molecules, potential drug-like molecules, using the mathematical formula of Gomberg's function, which relate the biological activity with the time or concentration of the molecules. So against STAT3 inhibition. So this is the process I just mentioned, and some of the example highlighted molecules which are reported, which having the activity against cancer cell, anti-cancer activity. You can see IC50 already mentioned here in the below. All are predicted through experimental assay and found that predicted activity and experimental activity are almost similar. So this is the flowchart of the method which we use for developing the relationship model. It's an in silico assay replacing the in vitro assay actually. This is also reported, and at the end we got that these ADME plot. You see, egg plot is also called the egg plot, which shows that whether the compound predicted actives are having the good ADME properties or pharmacokinetic properties or not. So those molecules point which are shown in blue color or purple color, you see these molecules are lying within the uncircled regions, which shows that ADME properties, they are very well followed and it's almost similar to known drugs. Similarly, further we go for biological activity of glyceric acid molecule, also called as 18-beta glyceric acid, which is a triterpenoid molecule, again anti-TB activity. And 9 derivatives of these molecules we have reported with the predicted as well as in vitro activity data in this journal, Current Topics in Medicinal Chemistry in 2015. So you can explore. Similarly, we reported the activity of the QSAR model based data through validated with in vitro assay for uracilic acid derivatives and against antiplasmodial activity. And this model is already reported with 96% prediction accuracy. You can explore more information about 8 derivatives of uracilic acid which having the good activity. And out of these 2 active derivatives, UA18 and UA21 show significant dose-dependent antiplasmodial activity against Plasmodium falciparum. So it's an antimalarial compound. You see, we have explored it and reported it and used in the formulation, herbal formulation for further studies for human welfare. So this data is already reported in RSC Advanced Journal in 2015. We have published it. You can explore more information about this in this journal. After that, ADME, we required to see the drug likeness score as well as ADME plot and toxicity related studies. These are the component which comes under the screening process. Preliminary screening required before going for clinical trials or the animal testing. So you must evaluate your compound after QSR that whether the compound lying within the applicable domain or not, as shown in the present slide. In the peak, within the peak, blue peak, which is a blue peak indicating the drug-like peak molecule. So you see the molecule as shown in blue color line, which is a glyceric acid molecule lying within the blue and under blue color peak, which shows that it is a drug-like molecule. So this way you can give the confidence that the prediction is good enough and it has good drug-like properties and good ADME properties. And within the egg plot, you see GA is there and comparable to chloroquine, which is a known control. We have used, you see both molecules lying within the applicable domain of the ADME plot, which is a pharmacokinetic related part. And then go for toxicity related studies, calculate the hepatotoxicity, blood-brain barrier, aqueous solubility, cytochrome 450 binding, plasma protein binding, and polar surface area. Polar surface area must not include like more than 140. You see the compound X12 indicating 144 PSA, 144 angstrom inspire, which is a polar surface area. It is crossing the limit of the standard drug-like molecule, which is 140. So 4 unit is, you can revise the or optimize the lead and further calculate the properties. So the first molecule is not good enough molecule. Rest are under the limit actually. Similarly, toxicity related, you can calculate as required by the different regulatory bodies in India as well as abroad. If you want to like approval of your molecule, you must show this data related to predicted toxicity with high doses as well as long-term use. So what are the different properties required to mention in the table? You must include the properties related with carcinogenicity. As I mentioned, these are the models available freely. You can use it. So I have used the TopCat, Discovery Studio TopCat. You can use Schrodinger or other softwares which predict the pharmacokinetic properties. And you see the second most important property is developmental toxicity. And the third is mutagenicity. And the fourth is skin sensitization. And the 5 is hepatotoxicity as mentioned in the earlier slide. So these are the 4-5 properties which you can use to calculate their properties in terms of values or quantitative or qualitative manner, and then compare with the standard drug. You see the first 4 column indicating the yellow color as highlighted, X19, X44, and X45 and 49. These are the derivatives compounds. And while in the last column, you see the known drug, doxorubicin, which is a known anti-cancer drug. So compare your properties with the known one. This is a very much required while submitting the data. This data is already published in 2014 in Drug Design Development Therapy Journal. You can explore more information about it. Further, you want to explore the mechanism of binding affinity, you can use the MD simulation. And for MD simulation, you must include NVT, NPT, and RMSD, RMS, and RG values and interaction energy data. These are the graph of the examples. You can use these information at least in your data so that you can get easily approval of your molecule for drug discovery or testing in human. What are the machine learning method used in QSAR model? As I mentioned, for high number of drug, you must include some machine learning method. Normally, we use the supervised or inductive method where we use training data set to develop a classification model or regression model. Normally, we use the classification model and regression model for prediction of the untested molecules. What are the aim of your study? As per your aim, you can select any one method at a time. So normally I use the regression method and go for multiple linear one. And for high throughput data classification, we use the KNN, random forest, Bayesian learning, ANN, artificial neural network, decision tree, etc. So based on your purpose, you can select any one method at a time. If you don't have any data or any machine learning information training data, you go for unsupervised method, where we used to use the clustering method like k-mean clustering and dimensionally reduction method, that is principal component analysis. And even the hierarchical clustering you can use to classify or cluster the molecules, small molecules, and go for model development further. So what are the steps in machine learning based QSAR models? If in comparison to 2D QSAR model, we use data collection, data pre-processing, model selection, model training, model testing and tuning, and the last is model prediction. So you must include these heading in your methodology section, which indicates that the model development is good enough and robust for taking any approval from the regulatory body. And now we have like discussed the 2D component, 2D QSR model. What do you mean by 3D QSR model? I briefly explain here in this slide that it is a different from conventional QSR approach in several aspects and form several advantages over it. In 3D QSR, the physical properties of each molecules are calculated by computer and no experimental constants are required. So you can go further without any experimental assay information. The most important part is you have to like calculate the properties of the molecule, overall molecule, not for individual substituents in this 3D QSR. So whole molecule information is required. While in 2D QSR, we also include the individual substituent information also. And the third is like properties known as in 3D QSR, we use the terms like fields. Fields means these are the chemical properties related with 3D structure. And most commonly in 3D QSR technique, we use the term steric fields and electrostatic fields. So steric fields define a shape of molecule, while the electrostatic fields define the electronic character of molecule. So these are the two important components we normally defined in the 3D QSR model. However, hydrophobic properties, for example, log P, log D, are less important in 3D QSR as compared to 2D QSR. So don't go for these properties. Here is the snapshot where we use the receptor complex information from PDB and go for pharmacophore mapping. So you see the uncircled regions indicating the steric properties and electrostatic properties, which regulate the biological activity. And these should be like similar in your queries at compounds. Once we identify these, we overlap it and find out the alignment to this pharmacophore structure component, and based on this pharmacophore, we can screen number of untested compound to find out whether which compound is whether fitting within the pharmacophore region or not. This way we can structurally identify the molecules. Here, the most important field developed first time is for 3D QSR descriptors representative is a group of descriptors basically, and this group of descriptors called as comparative molecular field analysis study. This is all in short. It is also called as COMFA and COMSIA related studies. COMSIA is this like further advancement within the COMFA. So not going in detail, but it's a group of molecular fields basically, which is first time used by Tripos Siebel software, and it include the like structural properties related to 3D QSR. It is normally used to build the QSR model or 3D model to indicate the regions that affect biological activity with a change in the chemical substituents. So the advantage of COMFA, if you see the ability, it's the most important, it's the ability to predict the biological activity of a small molecule. And the second is to represent the relationship between steric and electrostatic properties and biological activity in the form of contour maps. So contour map represent the 3D QSR. Wherever you see the contour map, you can understand that the author of the research group already done the 3D QSR. So I suggest you go for like 2D QSR first, then go for 3D QSR, which gives the key features on ligand receptor interaction and topology of receptors. So contour maps gives the information related with ligand receptor interaction sites and the topology of receptors required for binding of a small molecule. You see the snapshot I just covered, taken from the literature. For your understanding that you can identify in 3D QSR the best pharmacophore site within the molecule or training data set. And as highlighted, I mentioned here aromatic component, hydrogen bond acceptor related circle regions, spheres. You see, different spheres are mentioned here. Authors very well represented the aliphatic regions within the first diagram. In the second diagram on the top, we shows the alignment of the small molecule, different small molecules overlapping with each others, and it shows the conservation of the pharmacophore. Those components of the structure which are overlapping with each others are called as pharmacophoric regions, that should not be replaced or changed during the designing of molecule, while the unoverlapping part you can use for changing the molecule or designing the analogs by changing some functional group or elements. In the third diagram, you see, I just mentioned that the author represented the contribution plot of steric and electrostatic interaction in 3D QSR using the genetic algorithm partial least square method model development for use for QSR model development, where the alignment part indicating the training dataset compounds having overlapping with the two rings, and the third component is third ring and fourth rings are side chain. Basically, they are not overlapping, so they are not conserved. So in other way, we can say that the middle component basically is a conserved one. It's a pharmacophore region basically. And the last is once you align the compounds, you try to develop the plot using the regression method, and you find out the compare the predicted activity with the observed activity. Observed activity means experimental activity. It should lie within the diagonal line, and it is if it is showing the horizontal line, it means there is a no relationship. You have done a vague exercise. So you must develop the graph. It should show the diagonal line and the data points or the molecules lie within the line only. Training set and test set both. Now see the example of contour maps, how to visualize the contour map showing the hydrogen bond donor effect on a active molecule or inactive molecules. Here A is active molecules picture and the B is inactive molecules picture. So you see a contour map showing the hydrogen bond donor effect on the molecules. So different cubical structures are highlighted within the structures, with indicating the unconserved part and the conserved parts. So this way you can highlight the different steric and electrostatic properties within the structure using the sphere method or cubical method, or you can highlight the some chemical properties also within the structures if you found significant. So this approach is called as the 3D QSR approach, where we require the three-dimensional structure of the ligand as well as the receptor molecule. So explore PDB database and find out the complex, and you found if number of compounds more than 10 and 20, go for 3D QSR. So right now I don't find much complex structure in PDB related to a single type of drug category. So we did not go further for 3D QSR model development. But there is different approaches for developing the 3D QSR approach model. You can use that approach for for example, HQSR or over hopping method and so on, alignment method, and use based on your knowledge and training, you can develop the model. But I suggest you go for 2D QSR first and then go for 3D QSR. That will be a good idea. In this diagram, I just showed the contour map showing the hydrophobic effect of active molecule as well as inactive compound. So authors here represented the blue color component, which shows the hydrophobic effect are there. The molecule should have hydrophobic effect, otherwise it will be soluble in nature. So what are the limitation of the QSR? As I mentioned, there are some limitation of 2D and 3D QSR. So just for your information, just I highlighted few of them. The first is the calculation of these two main fields made it possible to study a larger variety of molecules than by traditional QSR. And the second is traditional QSR would not be able to cope up with the molecules having unusual substituents, since there would be no experimental constant record for that compound. And the third is QSR is restricted to molecule having same basic skeleton, whereas this is not the case of 3D QSR. So in 2D QSR, you must have the compound which similar in skeleton, while in 3D QSR, there is no restriction for predicting the query set compounds activity. And the last is both normal or 2D QSR and 3D QSR can be used to propose the new molecule for synthesis. So chemist uses both the method. It's up to you and your learning. You can join any group. But if you have an interest in like structural related things, structural bioinformatics, I suggest you go for 3D QSR. And if you want to do some work in relationship with chemists and biologists, you go for 2D QSR. So in the CSIR lab, I used to like go for 2D QSR, but in CDI, Dr. Imran Siddiqui is like doing the 3D QSR for lead identification. So you can use up to you. With this brief introduction, I just thank you. I say thank you to all for having listened this important talk. Thank you so much. Any question from the participant, you can ask.
Thanks, Dr. Feroz, for the very interesting talk. So I think we have quite a few questions in the chat box, so maybe I can
Yeah.
just read them out one by one, and then we can just go through the questions. So the first question is by Vichitra, who's asking, is there some example in which QSAR approach doesn't show any significant activity, but the preclinical studies show it?
Yeah, this has happened mostly because in database, if you see normally research student or the research scholars blankly having a faith on like online databases like Campbell and PubChem. But as you know that the same compound having a different activity in vitro assay by submitted by some other countries and some places. So if you explore in detail Campbell database, they shows the compound activity in different ranges basically, and like 10 micromolar to 20 or 15 micromolar for the same drug. We tested some of the VGFR inhibitors in CMAP, and we found that the reported activity of known drug is not similar to the standard activity which we found in the lab. So it means there is a wide variability within the reported biological activity by the research group. And they have made the databases and people are blankly using these databases information, which is not correct. I suggest you go for like research papers and literatures and collect the information of single compound known drug, whether which is the like minimum value, which is the highest value for activity. So always use the average value while developing the QSR model. I'm sure you will get the very good result at the end.
The next
Yeah.
question is by Manohar, who's asking, what is the difference between docking and QSAR model?
Docking is basically a simulation exercise without solvent background. It uses a force field for detecting the molecular interaction between the small molecule and the receptor. There are only two components. While in MD simulation exercise, it shows the binding affinity along with the stability. So if you want to find the stability with time, it includes the binding affinity information with time in MD simulation. Molecular docking and molecular dynamic simulation both are simulation exercise. But preliminary, we start with the molecular docking to find out the interaction between these two molecules, the small molecule or the receptor. While MD simulation, we use to detect the binding stability of the molecule with time, like in nanosecond or like normally we go for 100 nanosecond. So it gives the idea about the like behavior of the molecule, small molecule in the receptor, along with the solvent information. That is a good. And that's why MD simulation takes the time, almost 3 and 4, 5 days for single molecule complex simulation. QSAR is basically for predicting the biological activity. Okay. To make the relationship of the compound, it's a lead identification mechanism related thing. So we did not like cover it in the form of like receptor identification. So in drug discovery, there are four components. One is lead identification and lead optimization. These two components basically done by the chemist and the computational chemist. And nowadays, cheminformatics or the bioinformatics are allowing. They are doing in a team manner. So lead identification optimization is chemistry related thing. So you have to use only biological activity reported, and rest of the thing is like calculate the chemical properties and making a relationship for for using this model for prediction of the biological activity. So in other words, we can say that QSR is a virtual screening method actually for lead identification and optimization. You can change the side chain and then go for prediction and find out the good active compounds. So virtually you can design 40, 50 compounds, and without any in vitro testing, you can screen 4 or 5 potential compound out of 40. So this is a virtual screening method, basically, QSR. While the docking and MD simulation, molecular docking and MD simulation both comes under target identification and validation part. This is the third and fourth step of the drug discovery stage. And after that, we go for preclinical studies and clinical studies further. So these are the 5 steps, which takes almost 6 to 10 years or 15 years. But nowadays, in using the computational approaches, for example, HIV/AIDS inhibitors designed within 4 or 5 years only, with the help of this computer-aided drug designing method, QSR and docking and simulation. So nowadays, very commonly used in all the pharmaceutical companies and research group. In abroad, like if you go for any European country or US, there is a regulatory concern already they have given. Without QSR, they will not consider any data. So it is a compulsory component. But in India, it is not compulsory yet. But I am sure in future, when the team aware with this computational technology, they will include that components. So that is the beauty. And this way you can save the life of the animal. You see, you can like screen thousands of compounds without testing on animal. So you can prioritize the molecules, like out of thousands, you only get 10 compound or 5 compound through QSR. And these 5 requires only testing on small animals. Otherwise, randomly testing number of compound by the chemist design, then randomly testing it and publishing the data in research papers, they mention that for single testing, it uses 30 or 40 mice for single in vitro, in vivo activity. So you see the sacrifice is like LD50, lethal dose 50%. So they sacrifice 50% population, like out of 40, they sacrifice 20 mice. So to calculate the LD50. So to avoid this sacrifice, you can use in silico method like QSR and docking to prioritize the lead and explore, come up with potential information and suggest in front of the committee that these are the potential molecules having a good relationship. And this way you can save the wet lab economic. Yeah, thank you. Any more question, Surat?
Question is by Deepti. Although you have kind of addressed this, how QSR assists molecular docking. So I think this you've already kind of addressed this, but do you want to add anything to it?
Yeah, normally in CMAP, we start with the group of chemists and the group of biologists, which are animal scientists basically, in clinical studies. So we do start with the QSR first and then go for molecular docking and MD simulation, because sometimes it happens that QSR models, if they have designed number of molecules, so we have to prioritize the molecules. Otherwise, randomly testing all the molecules is not a good idea. So to prioritize the molecule, we need QSR model first. And if there is a relationship between the molecules properties and the biological activity, based on like prior published data, so we use that component. From single lab, we have like 30 years or 50 years of research lab data in CMAP. So we use that 30 years of data in the training data set. And based on that previously done work, in vitro work, we develop the model and make the screening process and prioritize the 10 or 15 molecules only out of 100 and 200 compounds. And based on these 10 and 15 compounds, we go for further for molecular docking and MD simulation. So in docking, mostly not all compounds, but 90% passes the docking process. But MD simulation, they fail sometime, like 20% failed actually. So the process flow is just like that only. You start with QSR, then go for docking, and then go for MD simulation, and then go for in vitro assays. Or parallel, you can do the in vitro assay. It's up to you based on your money availability.
One person is asking, can we do any wet lab test like GCMS before the QSAR modeling for more precise targeting?
Yes, if the sample is plant, for example, herbal sample is there, or the you have some natural compound from other organism like antifungal compound looking for. So you have to like give the information about whether that compound is present in the sample or not. So for that purpose, you need to submit the HPLC data or LCMS data. Or if it is the volatile in nature, I suggest you go for GCMS data also. Yeah. So that is a preliminary starting point actually. You must submit that information, then go for QSR, docking, and so on. It's a phytochemical constituent identification process.
Okay. Himanshi is asking, so how do we create a regression equation?
You can create the regression equation using MS Excel also. There is a package within that, a statistical package there. You can explore it through Google. Within MS Office, in Excel sheet, there is a package called as data analysis. You can use that data analysis pack. You have to activate, and by default, it is not there actually. You have to activate it first by going toward the setting part and selecting it. And after activation, you can select the data analysis pack under the menu data analysis. And that components covers the regression, correlation, a number of statistical method already there actually by default in MS Office. You can use it and you select the independent variable, at least 4 5 independent variable and one independent variable. That is a biological activity. And independence variables basically considered as chemical properties variable. So these two parameters you can consider while doubling the model, and it gives the automatically give the equation and graph also. It's very easy actually. Yeah.
Right. One person is asking, how can one assess the target specificity of these QSR-based derivative compounds, or as there is a chance that it can also bind with other proteins? Can we also include pocket information to build QSR models?
Yes. In QSR, that is the beauty. You can use customized information in training data set. You can use docking energy also as one descriptor. You can use other chemical properties which you think that it is very much important and it should be included. By default, software is not included that component. So that is a good idea in QSR. You can customize it and develop the model and see whether the improvement is there or not. If the R square value is increases, means good enough, go ahead, no issue. Yeah.
Right.
Dr.
Next
Suraj.
question, yeah, what is the difference between QSR modeling and pharmacophore modeling?
QSR modeling, as I mentioned, that it only required the chemical properties which are mostly two-dimensional in nature and using the biological activity in the training data set compound. While in 3D QSR, it uses the 3D dimensional properties, for example, steric fields and electrostatic fields, along with the three-dimensional space. So three-dimensional space, normally we uses the sphere component or the what we call as contour mapping. So that is the three-dimensional QSR. Pharmacophore modeling is also called as just like it's a 3D, it is a component of the 3D QSR. It uses the conserved regions within the known compounds, known active compounds, see whether how many fragment within the known drugs are common in all the drugs categories, and that conserved region basically called as pharmacophore that control the activity. You should not change that component. So pharmacophore modeling is required if you have PDB structural files data information, like complex information. At least minimum at least 5 to 6 structural components should be there reported. So it shows the conformation information. So the small molecule conformation as well as protein conformation, crystallographic data information is very much required in pharmacophore modeling. So if you have, it's a receptor-based lead identification process, while the QSR, 2D QSR modeling is, or classical QSR modeling is, it's a lead-based identification process, not receptor-based. So in pharmacophore, there is a must requirement of conserved 3D structural features, which is present in the crystallographic database, which is also called PDB database. So you must explore first how many structures are present within the PDB database, and if it is sufficient in number, like at least my concern, it is like it should be 6, or you can consider 5. So if you have a 5 complex or 6 complex or more than 6 complex, good enough. So you can just overlap these structures with each others on the same binding site and see different compounds within the same target how they change the conformation, whether they are similar or different. It is assumed that similar kind of the activity is controlled by similar kind of conformation pocket, or what is also called as 3D pocket, binding pocket, and this should be conserved within the structural component of receptor as well as the topology of the small molecule. So we normally consider the conserved regions within the small molecule as a pharmacophore regions. So the conserved pharmacophore regions represent the conformation of ligand molecule, not the receptor molecule. So that is the difference.
Then we have one
Uh,
last question.
All right. So we also have Manohar asking us again to
Yeah.
clarify the difference between docking and QSAR, and they have also added how small molecules can be docked.
So you can as I mentioned, QSR for predicting the biological activity and relating the biological activity with the chemical properties. So it's a lead identification method, while the docking is target identification method basically. Predicted active compounds through QSR model normally go further for checking the binding affinity of selected ligands on selected receptors, whether they are able to bind on the receptor or not. Sometimes QSR predict that the molecule is good enough, but later we found that after docking that it did not show any good binding affinity energy. That is a binding score basically. So we normally reject that compounds. So it may be possible that there may be error in QSR model development or some other reason may be possible. So docking is target identification method while the QSR is lead identification method. And the second question is what?
How small molecules can be docked?
Small molecule. Okay. So small molecules consider the inorganic compounds also and organic compounds which is within the 500 molecular weight region, and they are basically uh, you can easily dock these compound through using the any docking softwares like Autodock is freely available. You can use that component. Vina is already freely available. You can use number of molecules parallel using the Pyrex software also. It is freely available. It uses the ligand binding ability on the selected receptor sites. So genetic algorithm method basically used to identify the potential site of binding. And if you know the binding site region through the BDB database, then you can use that information to find particular small molecule and that component. For that, you need to attend the molecular docking and MD simulation related training program. I think number of companies and research lab organizing the training component within the bioinformatic regions. Otherwise, you can also organize the workshop with small molecules docking on receptor site. It is a very wonderful idea to give the training to these students. Yeah.
All right. So I think we have a last question here, asking if we have metals in the compound, how should we proceed with the docking?
Hello.
Pardon, please repeat the question.
Deepti is asking if we have metals in the compound, how to proceed with the docking.
So in PDB, if the metal is there, you can exclude you can remove the metals before docking, because in docking program, you can select ligand and receptor molecule. So rest of the molecule considered as heteroatom and they should be removed, including water molecules. So I think there is no specific software which shows the binding affinity of metal because it's metal considered as a cofactor. So I suggest you first bind the small molecule on the receptor side and do the simulation exercise. There is a cleaning step in the first step, pre-clinical step. Yeah. There are a number of softwares available. You see, Schrodinger is a commercial software company. They develop the good algorithm module within the docking program, and the software name is Glide. Glide is the softwares which normally used for molecular docking, but Schrodinger is very costly software actually. So I don't recommend it actually. I suggest you to go for like Discovery Studio software, or you can use Tripos software also. You can use freely available softwares like Autodoc. I mentioned Autodoc, Vina, and Autodoc. They are freely available on Linux platform or MS command prompt. You can use it. And for chemical descriptor calculation, use Paddle softwares. It is freely available. You can download it in Windows or in Linux, both you can do it. And for visualization of the structure, you can use any freeware software like PyMol or Sys PDB viewer, and so on. There are so many ResMol even also. So if you use the free one first and get training, there are so many training modules available in the like online media. Just get trained and learn it. It's a 1 month exercise, I think, good enough for molecular docking and MD simulation. It takes some time, like 2 months, to be perfect one. But you can like it's up to you. Like if you have an interest, you can learn it. So first step is the cleaning of the ligand. You have to clean the ligand, optimize the geometry, and optimize the energy. It should not be in excited state. It should be in normal state before like having interaction with the receptor molecule. And similarly, the receptor molecule, which is a protein molecule, basically, it should be optimized or cleaned well before like going for docking studies. So remove the heteroatoms, which are, I mentioned, that like metal or water molecule, remove it. Even the known inhibitors, they bind there in the binding site, remove it that also. And only the protein component you should include in the beautification process. After that, you include the hydrogen atom. Hydrogen atom mostly missing in the crystallographic information because the crystallographic method of X-ray and NMR method both did not include the hydrogen atom actually during the 3D model development process. So you have to include that component and then remove the charges, and after that do the docking process. So it's a exercise. If you want to learn more, you just attend one workshop from CMAP. I organize each year at the end of the year. We organize workshop, 7 days workshop. You can attend that component. We'll discuss that thing. Otherwise, yourself, you have nearby your group is there. You can sit and give time to spend learn docking method. It's a very easy exercise method, and I don't think it will be a difficult part for you. Okay. So start with like software, some of the freeware softwares, Autodoc, Vina, Pyrex, and Paddle software, PDB viewers, like that. Thank you. Any more questions?
No, I think the last question also asked about some resources to learn about computational biology and molecular docking, but I think we've covered most of it.
Yeah, there are so many resources available online. I suggest you go for online modules like Campbell, EBI, NCBI. They have mentioned known resources. Just explore these web servers, international web server first online.
Right. Thank you so much, sir, for this informative webinar.
Thank you.
Our participants have interacted, and I'm sure they have liked it quite well. So for the participants, I'm sharing a feedback form for our webinar series. Please do take some time out and fill it. I would also like to inform all the participants that we have an ongoing virtual lab series. It's called Back to the Future. And here we talk about cutting edge technologies in biological sciences. So you can check out the links in the chat box. And I would like to thank Dr. Khan again for his time and for this webinar.
Thank you, Kadambari. Thank you for nice anchoring, and thank you, Dr. Surat, for organizing such kind of workshop. And I feel privileged to be part of this whole webinar series program. And I wish all the best for your group. Thank you so much.
Thanks a lot, Dr. Feroz.
Yeah.
Thank you so much.
Thanks everyone for joining us today. Thanks a lot.